In the last lecture, we talked about what goes on in the first reactions of photosynthesis known as the light-dependent reactions. We talked about photosystem 2 and photosystem 1 being used and came to the conclusion that during the light-dependent reactions, energy is captured from sunlight by light-absorbing molecules, and those light-absorbing molecules are located in the chloroplast, that's chlorophyll, and the energy gets transferred to electrons that enter an electron transport chain. The water molecules get broken down into hydrogen ions, electrons, and oxygen molecules, and the water molecules then provide the hydrogen ions and the electrons that are used in the light-dependent reactions. We also said that energized electrons, we're going to have two functions. They're going to provide energy for hydrogen ion transport, and they're added to NADP plus to form NADPH. And that flow of hydrogen ions through ATP synthase is what makes ATP. The products of the light-dependent reactions are oxygen, NADPH, and ATP. And then oxygen is given off as a waste product, which we need, and energy from ATP and NADPH is used to make sugars. So we're going to look at the second stage in this lecture, and it's called the Calvin cycle, sometimes called the light-independent reactions as well. And that's just because it's not necessary to be done in daylight time. So let's look at the Calvin cycle. This is a series of reactions, and it's going to use the CO2 from the atmosphere to produce a carbohydrate. This process is going to include carbon dioxide fixation, carbon dioxide reduction, and we're going to talk about ribulose, which is RUBP, and it gets regenerated in the process of the Calvin cycle, or the light independent reactions. Okay, let's take a closer look at what's going on. First of all, let's talk about the fixation of carbon dioxide. Now, why does it need fixed? Well, this is what's going to happen. Carbon dioxide is going to be attached to a 5-carbon RUBP molecule, and that's going to cause a 6-carbon molecule to split into 2 3-carbon molecules, so the 3PG molecules we talked about back in cellular respiration. RUBP carboxylase is an enzyme. Remember, ACE is enzyme, so it makes this whole process occur. And there's a lot of this carboxylase around because it's a slow enzyme, so we have enough of it to be able to get a hold of to use. Let's look at the reduction of carbon dioxide. Each 3PG molecule is going to undergo a reduction to G3P, and this happens in 2 steps. So I know it's confusing, the 3PG, and then it gets reduced to G3P, but that's what's going on in this reaction. The energy and electrons needed for this reaction are supplied by ATP and NADPH, and this is what we receive from the light reactions, those 2 products. This is showing the reduction of carbon dioxide. So we have 3PG, and it gets ATP, gives it some energy, and BPG is going to be changed to G3P. So the big thing is we have 3 PGs that become G3Ps, and ATP is going to be changed into ADP and phosphate, and then we're also going to have NADPH that's going to turn into NAD plus again during that reduction. This is how we regenerate RUBP. It's going to be used in carbon dioxide fixation, so it has to be replaced. Every 3 turns of the Calvin cycle, we get this regeneration occurs. A 5G3P or 3-carbon molecule is used, and it's used to remake the RUBP, which is the 5-carbon molecule that we've been talking about. And here we see regeneration of RUBP. The importance of the Calvin cycle, why do we need it? G3P, or glyceraldehyde 3-phosphate, can be converted to lots of other molecules that are needed. The hydrocarbon skeleton of G3P can form fatty acids and glycerol to make plant oils, glucose phosphate, which is a simple sugar, fructose, with added to glucose is going to form sucrose, which is a disaccharide that we see a lot in the foods that we eat, starch and cellulose, as well as amino acids. So this one hydrocarbon skeleton of G3P is so important for us to have all these things that help us to survive on a daily basis and make up our bodies as well as the other organisms that are around us. So it not only just keeps us going as a food source, it's how we were made up and what we were made up of. So I want to look at an extremely simplified version of what's going on during photosynthesis to kind of maybe get a broad overview so that we can look more specifically and it not be such a scary thing. So we have light coming in, and that's where the light reactions are going to occur. That's going to take in water and give off oxygen as a waste product, and it's also going to take in carbon dioxide in the Calvin cycle. So during the light reactions, we have NADPH and ATP that are formed, and it's a good thing those are formed there because those are what get used up, and the energy is used to make sugars, and they go back into the light reactions as NADP plus, ADP, and your phosphate group that came off of the ATP and released energy in the Calvin cycle. And our ultimate goal, of course, is to have sugar produced as well as the oxygen byproduct that we use to be able to live and carry on the way we do. This is one step above the last slide that you saw as far as difficulty and the area that you need to look at, the depth that you need to look at, the process of photosynthesis. When we see light coming in, again, the light reactions, because photosynthesis happens in the thylakoid membranes here within these stacks of grana. We have oxygen given off. Carbon dioxide is used in the Calvin cycle, so we do the carbon dioxide fixation and the carbon dioxide regeneration, and that helps to make the Calvin cycle go. And the products of the light reactions are used in the Calvin cycle in the light independent reactions, and then those are recycled as energy is given off, and our main product again is sugar.